Anyway, so welcome everybody. So Carol and I met last week to kind of talk about this, and we're just going to kind of open it up. Some of the things that we were thinking about when we first met was, what is the future of neurodegenerative disease, clinical care, research, social engagement, and even ethics. And what are the current gaps that we see here at UVA and stumbling blocks, and they could be either lacks of talent, infrastructure, resources that are limiting us from being at the forefront of neurodegenerative science and clinical care. So really trying to think broadly about where the field is going and how we can be out front. And then also to think about what are those metrics of success? And then always asking ourselves, is this going to make UVA's program in neurodegeneration distinct and novel? And then, how specifically will this usher in preeminence in neurodegenerative disease at UVA? So the kind of broad topics on that. So that was just what we came up with.
And so we encourage all of you to just brainstorm, put forward your ideas. At the end of this, we're going to go back to the main room and I will summarize. But the other thing, Amy put it in the chat, is there is that idea notes link. It is very important for us to put any thoughts we have in idea notes or to like other thoughts in idea notes, because that is one of the ways, presumably, that they're going to use to look to see how much enthusiasm there is for neurodegenerative disease and brain science. So please go ahead and just give us your thoughts. Anyone start?
Carol, it's Tim. So I think it was really a nail biter about whether we were going to get that NIH Alzheimer's Center. Now you're correct that we came really close, and I think it's actually worth looking at, since this is about from cells to society, looking at what their critique was, right? Because I think if we strengthen that is a key thing, and I think it was a reasonable critique, or somebody could make that critique fairly well. And I guess it just applies partly because we just pulled the team together and so forth. But I still think the critique was that there was not a sufficient thematic connectivity across cells, imaging, neuropsych testing, and data science.
So thoughts on how to move that forward?
So I guess so the plan is to go back in 2023, right? So that's I guess is that a starting point to know? And then will it be the same theme, or does it have to be a new grant? I guess that's another thing. I don't know if too many people even know what the grant was that went in.
So it was the Virginia Alzheimer's Disease Center, and the theme was focusing on rural participants. It is to create a cohort for research, everything from cells to society. And one of the things that we are good at here is at getting rural participants and people of color, in particular African Americans. And in all likelihood, we will go back with the same theme. That is a strength of ours. But I think what would strengthen it generally is to have people from all areas, from cells to society, doing work in that area, for everything from normal aging to neurodegenerative disease.
Hey, Carol, can I just respond really quickly? Another key aspect that you brought into the picture was the care, the dementia caregiver dyads. Right. It was really important, and that has a health aspect as well as a disease aspect.
Right, and I don't know how I could have forgotten that, Tim, but just so people know, the idea is to look at both people with dementia and their primary caregivers, who are often spouses, and primary caregivers are at increased risk for dementia themselves. So we're proposed to use that as a cohort for people who are at increased risk for dementia and watching them, and also changing the trajectory of dementia in the person with dementia through caregiver behaviors.
So just hearing about it now, I wonder if that's an opportunity for cells, in the sense of what is it about caregiving for Alzheimer's disease patients that makes them more susceptible? Is it the stress? And what is it about the stress? Is it because they're also having issues with sleep deprivation or intermittent sleep, or is it something related to, say, microbiome changes that are occurring in the individual that's being diagnosed, and how that mechanistically moves them into a higher risk? I mean, maybe that's an area where you could get into cells or molecules that would help.
Absolutely. And so we propose to look at sleep, but I think the microbiome is an obvious place to go. And that is, as you were saying, getting more to the cell level. Other thoughts or ideas on anything? Doesn't have to be about the P30.
So Taji, you can jump in. You got your hand up.
All right, thanks. Yeah, so I really love forming this around a P30 because I think it sort of shows that we've got cohesive ideas that are coming together, and we can show that we're going to have plans for continuity and sort of continuing to fund the program. So one of the things that I've been working on in the last year or so is just identifying people that have an interest in developing research programs in Alzheimer's disease. And the number of people here at UVA is quite high, of people that are sort of new to the field wanting to jump in. So I'm really curious, how we could help to, if 2023 is the year that we'd love to go back with a new application, what the science could look like. As we just identified, microbiome might be an interesting area, but I know that there are at least about 14 people at UVA developing research programs that were related to neuroimmunology and glia and neurodegeneration. And so I think this might be really, it's an opportunity to sort of expand or redefine what the science portion may look like. And I know that Alzheimer's disease research centers across the country tend to offer pilot programs and have thematic research that's supported. So, I mean, I would also love to hear about what we have planned for that. Will there be a brain bank? Will there be access to samples? Do we anticipate that there'll be pilot projects for people that are new to the field? These are sort of things that we could work on in the interim, and with some investment, we could really take to the next level.
And the answer to all of your questions is yes, that's in the proposal, but that's something that we could develop and should develop across our community.
Excellent.
Jeff, raised hand.
Yeah, hi. Most of you probably don't know me. I'm a yeast researcher in the biochemistry department, but so we study aging using yeast. And so one of my questions, and we've been actually interested, I had a conversation with George Bloom last week of translating some of the cellular aging work we're doing with budding yeast into Alzheimer's, basically a quiescence and maintenance of quiescence. And so that's why I was curious, that's why I'm here, is to see, are there plans for seed funding? So I'm putting in an R01, and I'm going to bring George on, but I wonder if there are any opportunities for someone like me, like a totally neuroscientist, to try to get into the field. So that's my question. It sounds like there is some interest in that, and I'd be very interested in it.
So to be clear, in the P30, there absolutely is, but maybe Grand Challenges is a place that we could get funding so that when, for this to happen, so that when we go back in with the P30 application, we already have these initiatives started. Does that make sense?
Yeah, so I get emails from the Brain Institute every once in a while, but it wasn't really clear how a total outsider could become involved.
We are an open and inclusive group.
Good to hear.
Hey Jeff,
Yes, John.
Yeah, a couple of points. So the AD supplements through the NIH, they're like 1 year, I think $100,000. That's one way externally. There's also Virginia Alzheimer's group. It's out of VCU, but they funded a bunch of UVA researchers, and they like to bring in new people to the field. And then there's actually a new investigator coming in January, and she works in Alzheimer's disease in flies. So using other model systems, maybe, you know, the two of you guys could kind of leverage each other's expertise in those model systems to move things forward.
Yep,
too.
Drosophil was on my radar screen, so that's good to know. I'll probably reach out to you, John, to see how I can reach out to the new person. But
yeah, and Barry Condren's the other person
yeah,
too.
we've yeah, great. Cool. Yeah, that's what NIA has a lot of money for Alzheimer's, and so I'm basically going to try to expand it. We'll put Alzheimer's into my grant and then maybe get a better pay line. We'll see. But I think it's a fascinating topic, and hoping to get involved. So just wanted to meet you all. Hello.
John, who is the Drosophila person who's coming?
Jada,
Thank you.
I'll put it in the chat. I can
Thank
put
you.
it in. Mhm.
I'll speak up if no one else is right now. I am an economist, so I'm a little bit of a fish out of water here. But I wanted to just kind of tune into this because I do research on the economics of aging. And some of it's related to Medicaid and some of it's related to long-term supports and services. And from what I hear, it sounds like you all may be talking about following a group of people in Virginia, which could be really interesting because Virginia has established managed care for Medicaid. And many people late in life, after they spend down their assets, are going to rely on the state and on Medicaid to pay for services. And by shifting from fee for service to managed care, there's potentially huge changes in access to caregivers, to providers, and in the payments incentives, and so on. And so, if there's any opportunity to kind of, you know, have that be a piece of what you all are thinking about in terms of linking to society, I just wanted to put that out there.
So that's great. I had no idea you were here. And one of the things that we are doing is looking at our, I have grants to provide care managers to our patients and their caregivers. And one of the things I have been charged with doing is figuring out how that actually saves Medicare money because of unplanned healthcare visits that are eliminated. So I look forward to talking to you about that.
Hi, Carol.
Oh,
I'm
go
sorry.
ahead.
Is
Go ahead.
someone else speaking now?
Sure. No, go ahead.
This is Meow Meow. I have been doing machine learning research in Alzheimer's disease for quite a while. And I just joined UVA 2 years ago, and I'm in the ECE and computer science department. So I'm just wondering that whether this program will provide some platform for engineers who does the research that's interested in neurodegenerative diseases to bridge us to communicate with clinicians. Because right now that I'm really looking for, so I know there are a lot of funding opportunities out there for NIH to provide fundings to people who like me doing the similar research, but I really would like to reach out to people who are interested at UVA School of Medicine. So I'm just wondering whether the program can provide us the platform to communicate.
Great. Who else wanted to speak?
Hi, Harry here. I just
Harry,
want to
go
make
ahead. Yeah.
a couple of comments. First of all, I think it's advantageous if we frame whatever we do in Alzheimer's research and neurodegeneration more broadly as aging, and not be exclusively focused on disease processes, but on the process of normal aging across the lifespan. And I think that includes, a lot of the interesting comorbidities that are age-related, such as pain, for example, auditory loss, social isolation, inflammation processes, metabolic changes, obesity, and so forth. So I think that would be advantageous. That's one comment. The other one, I really feel there is a need to consider having some level of chemistry, biochemistry, and really medicinal chemistry added, regardless of what we do, because right now, we're just, whether we talk about the biochemistry of protein aggregates or whether we're talking about development of novel compounds to interfere with, new targets, we really don't have those colleagues in chemistry. And I certainly want to have that being considered, and may actually help the AD Research Center as well to have that in place.
So Harry, do you think, so first I agree completely with your first point, and second, do you think the biochemistry, we have the biochemistry colleagues here, or that's something we need to increase?
I do think we need to certainly think about medicinal chemistry as something that we do not have, and I think there would be opportunities with the cancer center as well, because I think that, a medicinal chemist doesn't have to be specialized on, creating molecules that are specific for, let's say, targeting amyloid or amyloid disease processes. They can work in the cancer space just as much. But I do think for us to move towards translation, we need medicinal chemistry.
Thanks.
Yeah, I have a quick comment. So I was thinking, the overall topic is very big, and however, UVA, has a lot of strengths. So it would be really nice for the people from the medical side can, kind of boil down the medical problem into some biological hypothesis or platform requirement. So this way, from the basic science part, we can think, okay, so do I understand that hypothesis? Do I have a platform? Actually, maybe not even a single person, maybe multiple people will say, okay, as three of us will respond to it because somebody has a platform, somebody has some, biological, tools and so they put together. So breaking down a complex medical problem into addressable biological questions, I think it's going to make a connection, for two parts.
Do you have thoughts on a forum to do that?
I imagine a lot of disease problems, we could not do any experiments on patients. For example, let's say if you have a hypothesis, you need animal models. You can say, we want animal like this. We can test this, can test that. Then we'll say, oh, because animal model may have some more behavior, some more cellular, some more, cell interactions. Then some of us will know how to respond. Say, okay, I'm good at that part,
Can I say something? So I don't have this frame very well, but I'm just kind of sitting here thinking and, thinking how we play kind of to our existing strengths as well. A lot of it, we have our own research areas, right? And it can be often very hard to then fit into a box when there's a call for, a specific funding compared to, playing to our existing strengths. And, when I look at the people that I know here, all working on various aspects of neuronal biology, but very few of us in aging models or aged neurons. And I think, finding ways that we can work together to develop better systems, to work on, what we do already, be it immunology, be it, EPHIS, be it infectious disease, but have more model systems that we kind of combined our expertise to make these model systems so that we can continue working on what we know and what we have strengths in, but, in an aging system as well.
So an example of that is medical education curriculum has changed from studying diseases to studying systems. And it sounds like that's similar to what you're talking about.
I mean, yes, studying, I mean, it comes with challenges, right? Especially in neuroscience. I'm sure most of us who work in cell biology often dissect an embryonic or a postnatal neuron. And we're not looking at an adult or an aged neuron. That's very different. The same goes for animal models. I mean, mice don't really age. How do we create an aged mouse? It needs to be exposed to diseases. It needs to be socially active. Thinking of ways that we can have kind of new model systems so that we can really look at a healthy or a pathogenic aging process in other model systems.
Let me just make one provocative comment, and that is if we could simply dial back the process of aging by 5 years, we would reduce the burden of any kind of age-related pathology by 50% globally. So, if we could come up with a system like that, a moonshot idea to slow down aging as a whole, age-related illnesses would be, and certainly Alzheimer's disease would be much less of a problem. I know it's a crazy, complicated idea, but just want to put it out there.
So that doesn't sound like a crazy idea to me at all. That would be extremely helpful in terms of preventing Alzheimer's disease. And the behavioral things that we do now are geared towards that. It's changing diet, it's increasing aerobic exercise. It's all things that keep us younger.
I second that. So that's actually what we're doing with yeast cells, is we're looking at interventions, including caloric restriction, which you can actually do with yeast cells, and looking for small molecules. And so that's sort of how that would translate into looking at Alzheimer's and neurons, mice, Drosophila, C. elegans even. So I totally agree.
I think Anna's comment is certainly still relevant, that, aging in a mouse is not necessarily comparable to aging of a human, is it?
I mean, it does make me think of, say, I don't know, like Yale's animal facility, where you can have, mice that are in more of a natural environment as well as some kind of resource, so that you have something that mimics lifespan more. I don't know.
If I could jump in, introduce myself to everybody. I'm Chris Hiley. I'm a biomedical and chemical engineer. And not to divert from that topic, but just wanted to weigh in and voice that, there are some, you guys probably, a number of you may have met Kyle Lampy. I know he's really interested in neuroscience and applications of biomaterials to neuroscience. I also am, and I think something to maybe keep in mind is over here in C's that, there's a large contingent actually of soft materials people and biomaterials people who are interested in neurological problems. There's another researcher named Rachel Lettieri who's working on a neurological issue. And for me personally, I just wanted to I guess raise our flag and just say that we'd be interested in engaging. I'm interested in soft materials that can be vascularized and maybe with blood vasculature or lymphatic vasculature and that sort, and see maybe how cells and glia interact with those. And so would love to chat more with any of you about that. And I do think that there's that cohort of us over here who might be useful if we're thinking about in vitro models of certain things.
So in terms of in vitro models as well, I mean, what we have been doing is, trying to convert fibroblasts right directly into neurons. So we at least have it going in the lab, and that way you're not rejuvenating the cells like you would an iPS cell. So in some way you maintain some age-related signature in the neurons. And if there are more labs who want to do this, it's tricky to get enough cells, right? And it's time consuming just to start the process. But if there are multiple labs working, on diverse projects, but with similar tools, then
coming together to create kind of a bank of differently aged neurons. You know, we started to get a bank of fibroblasts from individuals from 0 up to 88 years old. But, you know, having other labs working towards this would be helpful as well.
One of the challenges that I'm sure others have faced as well is every time you put a student or a postdoc on an aging project, you know, it's just like a frightening idea because, you know, like, you know, you got to age your animals for, you know, 9, 12, 15 months. And you forgot to do that one experiment. And here goes another 12, 15 months. I think there's a huge obstacle to our research is that the current models that we're studying, at least in the rodents, demand so much time, it's so much money to maintain. And I know we're all duplicating this. Many of us don't even know that we're growing the same kind of animal colonies at an exorbitant cost. So at a minimum, we need to get better organized, you know, in terms of what kind of animal models we use and sharing those most effectively, and sort of really making a joint investment in having an aging population that we can study, whatever that may be.
Yeah, so I have a comment, you know, responding to Harry's idea. The point about aging research is, you know, oftentimes we focus on the age part. However, you know, it would be nice for early intervention and early detection. So that could be a very interesting area. We study what's wrong in the beginning for neurons and glial cells. And that research will, you know, pay off twice. One is where we could be unique. Second one is that really shorten the timeline of research. We don't have to wait for 2 years to study the animals. Maybe 6 months will be enough. That will save a lot of money and as well.
We are about to be put back into the main room. Thank you, everybody, for your thoughts. I will try to do justice in putting this all together. But it seems like we have a lot of people interested in aging and neurodegeneration.